Dear colleagues, dear friends, it's time to start our last session of the Ising lectures 2025, dedicated to century of the Ising model and 100 University of the Bells of Igor Yukhnovsky, the founder of the Lviv School of Statistical Physics. My name is Erwin, and thanks Yuri, I have the honor to chair this session together with my younger colleague from the Institute, Julian Hanchar. So I would like to ask Julian to supplement me whenever he wants to. And Professor Erwin Frey will start the session and continue his lecture entitled Emergence and Self-Organization in Biological System, the first part of which we had yesterday. Erwin, I invite you to take the floor, or more exactly, as mentioned, Reinhard, the screen is yours. You have 40 minutes, please.
Thank you very much for the kind introduction. So let me share the slideshow. Should see it now, right? Just to check.
Yes, it's okay.
So we left off yesterday saying that we do have a quantitative model that explains the physics of the system and its biological relevance. And I was asking the question, oh, doesn't it work? Sorry, it seems to not do what I want. Oh, here. Okay, so I asked the question whether there's some new non-equilibrium physics that could emerge. And just to remind you of what we had yesterday. So the important feature that I want to get across is that these biochemical reaction networks are off thermal equilibrium. They break detailed balance, and therefore they have non-equilibrium currents. And in our case, the non-equilibrium currents you see is from the cytosol, so the interior of the cell, to the membrane and back, and also between different states and different conformations. And we'll try to exploit this now to understand what's going on in terms of the non-equilibrium physics of these systems. So here is another system where I would like to illustrate an important phenomenon that should strike you. So I mentioned at the very beginning that pattern formation also happens in the C. elegans worm. There it defines a asymmetry between the two sides of the cell. And I'm going to show you now a simulation of this reaction network in two different geometries. One geometry is a prolate shape, and the other one is an oblate shape. And what you should focus on is how the interface between the two domains evolves over time. So as I start the time evolution, you see the interface changes, it orients. And the important feature that you should have noticed is that the interface length is always minimizing. Symmetry would not dictate these particular patterns. They're only selected if you, in addition, require that the interface length is actually minimal. And that's an interesting, striking phenomena, because if you think about pattern formation, it's based on diffusion reaction equations. That means the particles that you have in the system, they're basically forming a gas up to the fact that they show chemical reactions. So there's no pairwise interactions directly between the molecules, and still it shows a phenomenon that you otherwise only know from liquid-liquid phase separation or from other systems like foams that have interface tension dominating the physics. So what's going on? Why is there this apparent phenomena that you shorten the interface length? Let me remind you of what the physics of liquid-liquid phase separation is, or liquid mixtures is. So basically, you have a thermodynamic relation that says if you have an interface with a density jump delta rho, then there will be a connection between the change in chemical potential because of curvature, kappa is here the curvature, and the surface tension. And that's known as the Gibbs Thomson relation. And the reason for that on a physical basis is very simple, because if you look at the curved surface here on the left, if you look at the number of neighbors a particle has on an outward versus an inward bended surface, then you see that there's a difference. And the one which is outward bent is energetically unfavorable as compared to the inward bent surface, where you have more attractive interactions. So curvature in a system that has pairwise interactions is always going to zero in this case, and that's the effect of surface tension that straightens out interfaces. All right, that's the physics of systems that show pairwise interactions. That's my question now, is there a concept that's analogous to surface tension in systems that form patterns? And can, based on these tensions, if they exist, these non-equilibrium systems form supramolecular structures in the very same way as interacting particle systems? So supramolecular structures would be micelles, mixtures, foams, all the beautiful phenomena that are based on interface tension otherwise. So let me explain. If you look into such a system, now let's focus on the ellipsoids that we have here and make a cut on the ellipsoid. So look at the surface of the ellipsoid. And then you remember that we have this cyclic flux between the membrane and the cytosol. And that's what I'm plotting now here. So the blue line that you see is the density on the membrane. Here you have a high density in dark blue. There's an interface. It goes down to a low density. And in the cytosol, you have the opposite gradient going the other way. Now, the currents that I was already alluding to is such that you have a zone here on the back. So here, where the blue arrow is, on the back of this cytosolic gradient, where the proteins attach from the cytosol to the membrane. Then there's a gradient. That means there is cytosolic flux because of diffusion gradients. On the other side, on the front of this interface, there's the opposite happening. There's detachment of proteins from the membrane back into the cytosol. And again, there's a gradient in the cytosol that creates a diffusion flux. And you see there's this cyclic fluxes that happen close to a non-equilibrium interface. And these cyclic fluxes close to a non-equilibrium interface, they have important consequences. So let me explain now. So you take a projection. So again, here you have the membrane density, which is high. So here in darker blue. Then here's the interface. And then on the other side, you have a low membrane density. Now you should notice that if you have an interface of a certain width, indicated here by this arrow, that as you bend the surface, the inner and the outer part of that surface at the interface, they're either compressed on the inside or they are stretched on the outside for very simple geometric reasons. And because of this distinction in compression and stretching of the interface due to curvature, the flux that you have in this reddish zone will be enlarged because of the enlarged area, and the flux in the attachment part will be decreased. And the opposite will be on the other side here, where you have a curvature that's the other way around compared to this side here. So here you will have then a decreased detachment flux and an increased attachment flux. And that just means between the zone on the left and the right with the two different curvatures, you will have particles that effectively go into the cytosol, and that means you have a higher concentration here at a positive curvature, and you have a lower concentration here at a negative curvature. And then again, because you do have now a gradient, and this gradient will create particle fluxes that will even out the interface. So despite the fact that there's no pairwise interactions, the non-equilibrium fluxes that you have, they do create a non-equilibrium surface tension, and that surface tension simply is related to the area mismatch that happens as you bend a surface with some positive or negative curvature. So we do have surface tension here, and the reason is, as I explained now, the non-equilibrium currents that you have close to the interface. So we do have surface tension. Now let's ask ourselves, does that matter for the corresponding biological system? And here I show you data from the group of Petra Schwille. And those data were taken also with the min-D and min-E system for a certain parameter regime, where they show these kind of patterns as you see on the left. So that the main walls kind of are the min-E proteins in blue, and the inner area of these domain walls are the min-D protein in purple. And that looks like a foam, right? This looks very much like a foam. So let's check. Is this really a foam-like structure with all the mesoscopic features a foam has, and with all the laws that you know for the physics of foams? And there are quite a few. There's for Neumann's law, there's Neumann's law, there is the balance of surface tension, etc. And there are the plateau laws. So let's check those laws first. So the first thing you can do is you can make tripods here at the vertices and ask yourself, what are the contact angles between the different domain walls, the interfaces that you see here, and they are flat on, dead on, 120 degrees. So this is one feature of a foam. It has very nicely these 120 degrees angles. A feature that you don't have in foams is one that's seen here, namely that the foam here, this pattern-forming Turing foam, is not coarsening. So over time, it doesn't go to a state where the cells become larger and larger. No, it stops at a certain point in time where you then have an equilibrium distribution of domains with a certain typical area, which is here around 100 micrometer squared. And that's interesting, right? So you have something that's reminiscent of foam, the 120-degree angles, but the foam here doesn't coarsen in contrast to a liquid foam. What we can do now, and that's the upshot of the theoretical and experimental analysis that we have been discussing over the last lecture, is that we do have a fully quantitative model of the min-D, min-E system. That means what you see here in the experimental data, you can also study using computer simulation with a fully validated quantitative model. And that's what we did here. So we studied whether in a certain parameter regime, these min systems show these foam-like patterns, and indeed they do. This is an example of such a foam-like structure. The advantage of having computer simulations available to you is that you can do experiments which are hard to do in an experimental setting. So what we did here, that's Hendrik Weyer's work, is he took the foam in its equilibrated structure, that's now the size distribution that you have in the steady state, and then you compress the foam, or rescale, better to say, you rescale the foam to a size which is not the equilibrium size here. So this is too small. The foam has to coarsen to go back to the equilibrium state. And indeed, if you look at the simulations now, you see the foam over time coarsens and goes back to the same kind of equilibrium distance and size distribution that you had when you started with. With those kind of experiments, you can now study how the different cells that you have change over time. So you can study what happens to the cells that have 5 edges, what's to the cells that have 6 or 7 edges. And here is the data. You see that the domains that have 7 edges, they grow over time. The domains that have 6, they stay roughly constant over time. And the domains that have 5 edges, they shrink over time. And that's a famous law. It's the von Neumann law that one knows from the physics of foams. And the interesting part is this von Neumann law mathematically hinges only on the fact that you do have surface tension, And then the way you derive the von Neumann law, and I'll show this in a second, is basically that you use the Gauss-Bonnet theorem from differential geometry to show that the domains with the 5 edges, they must shrink. The ones with the 7s, they must grow. And those with the 6 edges, they stay constant over time. But now what's the reverse process? So as we saw, there's an equilibrium distribution of sizes. So if you do the opposite, if you scale up your system, you make it larger, then there must be some processes that bring you back again to the equilibrium structure. That cannot be coarsening now because it has to be the opposite. You have to go from larger cells to smaller cells. And indeed, when you do the simulation and let it run, you see there's kind of cell division phenomena, domain splitting, that happens and brings you back to the equilibrium structure. And those domain splitting events, they are topological transition in these foam-like networks. And that means you have now a balance between coarsening following von Neumann's law and the anti-coarsening that's dominated by inverse topological transitions. I mentioned the von Neumann law, so let me explain how this comes about here. We know from our very general considerations that there must be surface tension in the system. So the interfaces are driven by those surface tensions. And that means the normal velocity at the interfaces has to scale like the curvature. And now you can calculate what the time evolution of a domain actually is. You can calculate the time derivative of the domain volume, which is the integral over the boundary times the velocity. That gives the change of this domain volume. And that's then because V perp is proportional to kappa. This is the boundary integral over the curvature. And that's where the Gauss-Bonnet theory comes in, because the Gauss-Bonnet theorem tells you if you integrate these domains over the curvature, you should get the number of edges of this domain -6, and that gives you the time evolution of the corresponding area. And that's exactly what you see. That's the von Neumann law, and it's nicely fulfilled also for these non-equilibrium pattern-forming systems. So let me continue. Now let me show you the data before I run it. So this is again the experimental data from the group of Petraschwili in Munich. And I've picked now certain regions. So this region here in space, where you have a five edges, a domain here with these 5 edges, as you see. And then there's another domain where you do have an initial domain splitting event, kind of starting out here. Let me now run the experiment, and you see indeed this domain with the 5 edges shrinks over time, and here you have this domain wall forming this cell division or domain splitting event happening in the experimental data. All right, so there is indeed a foam-like structure, and maybe this is a good point now to summarize this part of my lecture. So yesterday, I told you about pattern forming in mass-conserving diffusion reaction systems. And what we have accomplished over time is that we can understand the pattern formation in these systems in terms of a local equilibrium theory, where the mass redistribution is the driving force shifting the equilibria in space and in time. Today, in this short fraction of my talk, I've shown you that these protein patterns can actually not just form biological patterns with a biological functional relevance, they also form new non-equilibrium patterns that we termed Turing foams because they are dominated by a non-equilibrium interface tension of the domains. All right, so that's the part on pattern formation. And then I was thinking of what should I tell you about today. And I thought I might tell you something about foams not in pattern-forming systems, but forms in active matter. So let me start. This is to remind you of something very fundamental, which I'm sure you know very, very well. If you think about the thermodynamics of phase transitions in equilibrium, then a very important concept is a phase diagram. And the next important concept is that of an order parameter and of a control parameter. And those two things do have a very distinct role. Order parameters, like the magnetization in a ferromagnet, or other things like in liquid helium or in liquid-liquid mixtures, these order parameters are dynamic variables. They're dynamic in space and in time. The control parameters, they are distinct from that. So here in a ferromagnet, the control parameter, for example, is the temperature. It's externally fixed and you can tune it, but it's not variable in space and time unless you study some particular systems where you have randomness in your system. All right, so that's the distinction. Order parameters are dynamic variables and control parameters are fixed but tunable, externally determined variables. And that's not just true for a ferromagnetic phase transition, it's also true for binary mixtures and many other phase transitions that you find in equilibrium thermodynamics. What you can ask now is how much of those concepts survive when you go to non-equilibrium systems, to active matter systems, for example. And the paradigm for that is nicely illustrated by this movie here. I guess the switch. So all right. So I had to turn down the volume because there's some voice in the movie. So I wanted to illustrate here with this movie is that patterns formed by birds or fish or any other kind of self-propelled particles, organisms, or whatsoever, they show beautiful patterns, which are patterns that are formed because these systems are far from thermodynamic equilibrium. And I want to study those now in the context of a biological process. And this biological process is the formation of patterns in the cytoskeleton. The cytoskeleton of a cell is composed of two different things. Basically, it's composed of biopolymers. These are these green objects here. Those are stiff, long polymer filaments, varying in length between a few microns and 10 microns. And they are responsible for the cell elasticity, but they're also forming tracks for molecular motors. So as you see here, they're kind of highways, and molecular motors, they can attach and detach from those highways. And once they're attached to the highways, they move from one end of these filaments to the other. And that's here nicely illustrated by an experiment from the group of Joe Howard that shows how these molecular motors move along such a microtubule. The speed of those motors is quite significant. It's from 0.02 up to 2 micrometer per second. And the forces they can generate are significant. They are in the piconewton range. And piconewton is really comparable here with the thermal energy scale that you have at room temperature or body temperature. So what we wanted to understand is, can you get complex behavior like the ones that you saw with the birds, also from these biopolymer systems? Because it's relevant, right? In the cell, you have lots of structure formation, and you would like to understand the physics behind those pattern-forming systems in the cytoskeleton. So what's shown here is a cartoon. You have the microtubules, two filaments. They are connected by molecular motors, and they exert relative forces between those microtubules. So they can slide them with respect to each other and also apply torques in different directions. That means what you have here is a combined systems of filaments and motors. The filaments are such that the motors orient them. There's mechanical work. And the filaments are also such that they transport the molecular motors along their tracks, and thereby they act as transport channels from one region of the many-body system that you see here to the other region. So let me show you now data. These data are from Zvonimir Docic's group at UC Santa Barbara. He put together many of those microtubules and many of those molecular motors and then studied what kind of structures do you form. So here you see at a low microtubule density, you see the formation of aster-like structures. So the filaments are here sticking out in the aster-like form, and the molecular motors, they are forming condensates here in blue inside of these aster-like structures. When Zvonimir and his group increased the density of the microtubules in the system, so the polymers, the filaments in the system, what they saw is the formation of foams, and these foams are quite dynamic, as you can see here from the movie on the left. They self-organize into structures which are foam-like, and then the steady state, as you see here in this picture, has really these interfaces where blue means kinesin 4, that's the molecular motors, and the black indicates the microtubules. So what's the physics here? Why are these mixtures of microtubules and motors self-organizing into these foam-like structures? Well, let's try and do some theory and construct it bottom-up. So from the basic interactions between the motors, try to make a kinetic theory, and then this kinetic theory is coarse-grained to ultimately derive a field theory. The basis is the interaction between the motors. You have two filaments. The motors can slide them with respect to each other because they exert forces and torques. And you can idealize this kind of interaction in 2 dimensions, that's the system we look at, by a collision rule. So you say you have these filaments, they have a certain orientation indicated by the arrows here. And if the angle of collision between the two filaments is less than a critical angle omega c, you say they align in parallel. So you have ferromagnetic order. And if they collide at a direction which is larger than this critical angle, they anti-align. So they form kind of an antiferromagnetic local order. And on top of it, once they have this anti-alignment, they can usually not fully move relative to each other. They remain in a state where there is an overlap region between the two anti-aligned polymers, and this overlap region is characterized by this parameter eta. It's basically because the motors show crowding phenomena, and that crowding phenomena inhibits the complete motion of the two filaments such they don't have an overlap region. Sorry. Please, if you have questions, interrupt me anytime. Now, how would you do a theory for that? You do a kinetic theory, and this kinetic theory is constructed such that you write down a differential Chapman-Kolmogorov equation or Fokker-Planck equation mixed with a master equation. What you say is that probability distribution function of the angles between the different filaments is a rotational diffusion term. And then you have gain and loss terms because of the collisions that you have here, the aligning collisions and the anti-aligning collisions. And I spare you here from the mathematical details. You can analyze these equations, and from the equations, you can determine a phase diagram. And this phase diagram shows here what kind of order do you get as you change the critical angle omega c. So you change the angle omega c between 0 and pi. And if you have large critical angles omega c, then you favor the polar alignment. If you have small critical angles omega c, you favor the anti-alignment. So you get here a phase diagram where you have polar order or nematic order, or in the language of ferromagnets, you do have something like a ferromagnetic order, and nematic order would not really be antiferromagnetic, would be something which is more like a liquid crystal, where the orientation is kind of random and both directions are equally likely. All right, so that's the phase diagram you get, and we put ourselves in a region where you primarily do have a polar order.
May I ask a question?
Yeah.
I mean, what is the starting situation? I mean, if you have all angles, you must have a symmetry. I mean, the angle omega, you have all values of omega at the beginning.
At the beginning, you have a homogeneous mixture and kind of disordered system. That could be the initial state. And then you ask yourself, as you change the density, so here the other control parameter is density, is this homogeneous state with disordered orientation, is that stable? And what this phase diagram shows you is that there's spontaneous symmetry breaking, and the spontaneous symmetry breaking is either to polar order or to nematic order. Okay, so ferromagnetic order, if you like, and this nematic phase transition is one where the filaments align, but they are still disordered with respect to a center of mass position.
But
And homogeneous state is white, right? On the phase diagram.
White means disordered.
Disordered, right?
So if you, for example, if you fix your critical angle to be omega c equal pi over 2, it would mean as you increase the density, or here you have the density, you go up here, you completely disordered, and then you have a phase transition with spontaneous symmetry breaking into nematic order. And if you were to go here with omega c 3 pi over 4, then you have also a phase transition, but now it's from disorder to polar order.
And omega c is determined by eta?
The omega c is a model parameter. It will depend on the microscopic details of the interaction between the microtubules and the motors. That's something which is not part of the theory. It's a parameter that we use in order to be able to describe different types of interactions between the motors and the microtubules. So in the end, if you would map the theory to the experimental data, you would find a particular value of omega c and a particular value of eta. They are indeed like, what could I take? Some molecular feature that you would have in a liquid-liquid mixture, like the interaction strength between the molecules. That's how you should think of the parameters omega c and eta.
Thank you.
Okay, so I hope that clarifies it. So now, in addition to this pairwise interaction that favors polar alignment or anti-alignment, there is intracellular transport, and that means that the motors which facilitate the interaction, right, they're responsible for this alignment and disalignment, they are also transported along the microtubule. And that means the density of these motors, m here, is not a constant. It will be linked to the order. So P is the polarity, and here that term is a drift term. That means you have motion of molecular motors along the direction of the microtubules. And on top of that, if the motors are detached from the filaments, they randomly diffuse in the environment. And that's kind of crucial because this allows you to change the interaction between the microtubules because the motor density is the parameter that determines the strength of the interaction. If you were to take an analogy to magnetic systems, what this means is that you have a dynamic variable that changes the exchange interaction between the spins. So now let's construct the theory. So on the left, I again show you the interaction as we have it. So you have some kind of alignment or anti-alignment interaction. And now you write down the Boltzmann equation for that. And don't show that. This is a lengthy equation that shows the kinetics of the variables that we have. And once you have such a kinetic description, you can, by coarse graining, really by explicit coarse graining, defining the density, the polarity, the nematic order parameter, and so on, derive a field theory. And I'll guide you now to the field theory that we derived bottom up from these underlying microscopic rules. So there's a first equation. It's a scalar term. It's a scalar term for the density, rho. And if you look at it, it looks complicated, but it's not that complicated at all. So the density changes over time with a term that's proportional to the gradient squared. And the first term that you see here, d rho, is nothing but diffusion. So microtubules basically diffuse. The other terms that you see are Taylor expansion in rho, so rho to the second power or third power, and they account for the fact that diffusion is also changed by the action of molecular motors because they randomly push and pull on the filaments, and that leads also to an effective diffusion, but that depends on the motor density m. And those terms here, they can represent something like crowding because you have high densities of the microtubules next to each other. The second line here is a different type of term. It couples the density to the polar order parameter. So pi is the vector for the polar order, describing the orientation of my filaments. And here pi, pj means that you have a tensor structure, and p squared is just the magnitude of this polarity here. And what these terms mean is if there is some kind of polarity in the system, then you have polarity currents that also change the density of the microtubules. So you have an anisotropic current because of the polarity in the system. The equation for the polarization looks even uglier, but again, it's not really complicated. The first term that you see on the right-hand side is a term that you would also write down if you would model a ferromagnet in terms, or Ising model in terms of model A. So this term just means that you have a linear term. This is the mass or the reduced temperature. It can change sign, and as it changes sign, the potential landscape changes to a Mexican hat potential. And that here just is the phi to the fourth term that you do have in a model A type of dynamics. So this first term here is nothing but the ordering term that you would also have in a model A description of an Ising model or a Heisenberg model. The second term that you have here are also similar to what you would have in a model A dynamics. They are coming from the fact that as you have gradients, the gradients will lead to some effective elasticity. There are more terms which are allowed here because you don't have the nice symmetry that you have in a Heisenberg ferromagnet. You have less symmetry, and that's why you can rearrange the gradients in different ways. The second line here is a coupling now back, a feedback to the density and also to the advection terms. So here you have terms which are proportional to the gradient in rho. And that means, for example, that the temporal change of the polarity will anchor along gradients in the density or gradients in the polarity. That's why I call these terms anchoring terms. And those terms here, they are like in Navier-Stokes equation, they're P nabla P terms. And that just means because of the relative sliding of the filaments, you have something like self-advection. Right? So all these terms have very natural explanations. And what you should always notice is if I indicate something in green, blue, sorry, in blue, that means that these parameters are depending on the microtubule density, on the motor density, sorry. And let me just note this term here. So here it means that the reduced temperature term has an explicit dependence on the motor density. Also, the strength of the 5⁴ term has an explicit dependence on the motor density. And that means really that the kind of order you have, whether you're above or below the critical temperature, here the critical density, is a function of the density of motors that you have here. So it could mean in some regions of space, the system wants to be polar ordered, and in other regions of space, it's in the disordered regime. Okay, and finally, this is a simple equation. It's just the diffusion advection equation that I already showed you. Motors diffuse if they are not bound to a microtubule, and if they are bound to a microtubule, they are advected along the microtubule. So that's the set of equations that you have and that you need to solve in order to understand of what's going on in these interacting many-body system with filaments and molecular motors. Okay, what do you get? You can do lots of different types of analysis that would take a long time to explain. You can do a linear stability analysis. You can ask under what condition is the spatially homogeneous state stable and when does it become unstable. That gives you certain boundaries in the phase diagram. And here, let me just jump right away to the findings. But before I do so, let me allude to one thing which I think is rather general, and let me explain it to you. So what do I think is general? Namely that the distinction between order parameter and control parameter that you have in equilibrium is no longer valid here. It's kind of broken. This dualism is different. In our case, the density is a control parameter, but it's also a dynamic variable. And as I explained just on the previous slide, because it's a dynamic variable, it changes the strength of the local interaction, so it leads to local symmetry breaking and not just to global symmetry breaking. The polar order, which is actually the order parameter, transports the density. And in that sense, the order parameter also becomes a control parameter as it changes the density. So it has an effect on the control parameter. So in all, there's a dualism between the two, control and order parameter. They don't have fixed roles anymore. It depends on the context, whether something acts as a control or an order parameter. And that interplay, I think, is the underlying meta reason why you have those kind of beautiful phenomena that I show you next now. So here, when you simulate or solve these equations numerically, using some standard finite element solver, you find an extremely rich phase diagram. And when we saw this the first time, we were really amazed what kind of structure you get. So here, what you change is the average microtubule density. At low microtubule density, we see what Zvonimir Docic is seeing in the experiment. You see these asters or micelles, where you have a polar order of the filaments in red here, surrounding a region in blue where the molecular motors are focused and concentrated. So you have a supramolecular structure maintained, again, by non-equilibrium currents. As you then increase the microtubule density, you find different phases. So here, an indication, you find bilayers, where now you have the filaments pointing inward into a zone where you have a density of motors, again in blue. So you see here, this here as well. That's the density of microtubules. This is the density of motors. You have these star-like structures, and each arm of a star corresponds to such a bilayer. And as you go further and further in microtubule density, you finally arrive here at a situation where the structure is really very reminiscent of what you have in the data. namely a foam-like structure where the microtubules form the boundaries of those, again, with the sandwiched molecular motor density in between. Let me show you how this works dynamically. This were just still images. Here you see movies where the dynamics, you can clearly see the dynamics of it. The upper model is the one where we strictly derived it from the molecular details. So this is really from the microscopic interactions all the way to the field theory. And it shows phenomena which are quite reminiscent of the data, but not quite. As you see, there's some kind of branchy structures that you don't see in the data. So we took the freedom that these field theories, of course, can also be based on symmetry and tweak the parameters a bit. And then you see this lower part where you see this nice foam-like structure with a coarsening and the dynamics that clearly shows that you have here the formation of an active dynamic foam. All right, so that's what I wanted to show you, and maybe that's a good point now for me to close and summarize the part on Turing and active foams. So in the Turing foams, you get interface laws for reaction diffusion systems, and you can use concepts from differential geometry and the fact that you have non-equilibrium circular flows to derive those interface laws like the von Neumann law and the Neumann law. And in active foams, you saw that these active foams, they show also supramolecular structures, just as equilibrium structures do, but it's not lipids that self-organize, it's actually motors and filaments that self-organize. So in a way, you can think of these systems as amphifiles, but as active amphifiles. All right, and here is kind of a cool outlook, just to get you curious. I started with these E. coli cells. I told you something about Turing foams, and here, this is a recent experiment from the Schwille lab that puts a vesicle on top of such a Turing foam. And what this vesicle does, it shows cell-like motion, but without any kind of molecular motor, without any kind of mechanical forces in the sense of something pushing and pulling. This is just by the interaction of the patterns with the geometry of the vesicle that gives rise to this cellular motion. So let me stop here, I guess, with my talk and give also thanks to the people who have done the work. So I want to mention in particular two students. One is Hendrik Weyer. Do you see my slides still? Sorry.
No.
No, I didn't share.
Pardon? You have to share?
Yes.
Okay.
Yes, you stop sharing. Mhm.
Okay.
Just say.
So that's an important slide. Now you should see it. So I want to mention two students in particular that have done a tremendous job. So Hendrik Weyer, he's just about finishing his PhD. He's the student who worked out the Turing foams and came up with the concept of line tension in these non-equilibrium systems. And Filippo Di Luca, who is now doing his PhD in Cambridge, he's the student that determined the active foams and their behavior. And of course, I also want to mention my experimental colleagues, Petra, Nikta, Kes, Lidevai, Sukyun, and Andreas, because without them, we would not be linked to reality. And I really am thankful for their ideas and their beautiful experimental data. And thank you for your attention during my talk. Thank you.
Thank you very much for your very nice presentation and very interesting talk. We have, in fact, not so many time, just a few questions. If you, so colleagues, please. Lucas, please.
Yeah, thanks for the great talk and both talks. I have a question about these min oscillations. So I first heard about them in a talk a couple of years ago by Robert Laughlin. I think he also got interested in these oscillations, and then I talked to some biologists because I understood that this might be one of the mechanisms leading to cell division, right? Which is associated with identifying the center point of the cell. And then in conversations with biologists, it sounded like that the actual mechanism is much more complex. And so I'm still unsure if these models are able to predict cell division. Maybe, it would be nice to hear your opinion on that.
Very good question. So of course, I mean, biology is complex and we try to approach it stepwise. Indeed, the min patterns are important. They're very important for cell division. If you take away the min oscillation, cell division doesn't work properly anymore. So they're really responsible for placing the division plane at midplane. What it doesn't explain is how cell division happens, because it just is a template that focuses another protein machinery made out of FTSZ polymers. These are like the microtubules, similar kind of biopolymers. And those FTSZ polymers, they are guided to midplane position by the min pattern. And once they form, they build the cell wall. And by building the cell wall, they divide the E. coli cell into two. So the role of the min pattern is to form a template, a reference frame, a coordinate system on which a secondary molecular process happens that makes cells divide.
Great, thank you. That's very helpful.
Thank you. So Yuri is the last question, please.
Yeah, thank you very much. Thank you very much for coming and for giving this talk. A short question with respect to the phase diagram you were showing. If I think about it as about usual phase diagram, am I right that the transition which is from disordered state to the polar state would be the second order transition? Then on the left it would be first order transition, and in between this omega C was showing us kind of tricritical point?
Not quite. Most of those transitions are actually first order.
Right.
And what happens is that as you change from disorder to polar order, on top of it, the polar order shows phase separation, meaning you get bands of polar order and no density in between. So it's not a second order phase transition in the sense that it would have critical fluctuations. It's more like a first order phase transition close to a binodal or spinodal, where you have on top of the orientational order, you also have demixing that happens.
Okay, thank you.
You're welcome.
May I ask you another question just very shortly?
Please go ahead.
I mean, you have a combination of order parameters and variables and the control parameters in your equation. So this is an extension what I just name model F or something for superfluid.
Yeah.
And do you did you look at, so to say, solid state or condensed matter physics systems where you can apply them out of biology? So I could think of a liquid mixture or even superfluids to apply it.
Yeah, I didn't think about superfluids, but you certainly can think about different types of liquid mixtures.
Yeah.
Where similar type of field theories might apply. What's different though is, and that's quite important, if you wanted to write the theory that I have in terms of gradients of a free energy function, or some version of it, so in the classification scheme of Halperin and Holmberg, you cannot. So there is no underlying free energy landscape that you can write down. That makes them very distinct from a thermodynamic system. And that's also the reason why you do have all these beautiful, phenomena with coarsening arrest, foams, micelles, etc. So in that sense, they're really different because they are non-gradient systems. Beyond the case that you have mode coupling, but really non-gradient in the sense that you don't have free energy functionals here.
Okay, thank you.
Do we need break, or we can continue because we are a little bit out of the program? Yuri?
I think we are good with respect to program. Let me check. The next lecture should start at 4 o'clock, and still we have 5 minutes before the start of the next lecture, right?
Okay.
Correct. So we have now a small break, and then Romualdo. You can,
Okay. We will
Again,
you can try and share
4
the screen.
o'clock in Lviv.
4 in Lviv and 3 in some other places, and maybe some other time.
That's correct.
Yeah.
So again, from my side, thanks a lot. I was very honored to give one of these Ising lectures. Thanks for invitation, and best of luck for the future in the Ukraine. So
Thank you very much for
Okay.
coming
next time
here.
I hope to be there in person, so if possible.
We are looking forward for our future meetings. Thank you.
Yeah, thank you very much.